Good morning, everyone. I'm Aksa Igzeneza, an assistant lecturer at the University of Rwanda. I welcome all of you in this second session uh, of seminar, which is prepared by the Inst Africa. Inst Africa is actually innovation, science, and technology for action and learning in Africa. So I'd like to just give our brief background on the Inst Africa. How did it come to generation? Before we start with our second session. So normally Install Africa started in October 2023 in Rwanda. And this happens due to collaboration between different institutions, which are the FIRAT, Foreseen Insight of Research and Translation Rwanda, and CST, which is National Council for Science and Technology in Rwanda, RBC, which is Rwanda Biomedical Center, NIRDA, National Industrial Research and Development Agency, UR, University of Rwanda, FDA, Rwanda Food and Drug Authority. So all those institutions worked together to, to make this Install Africa happens, which aims in the use of Drosophila, Mironogaster, fry model, and infrastructure for enhancing fundamental research capacity in Rwanda. So normally now, with this big project, which is being in Rwanda now, we are going to have sessions, which the second session on Drosophila and Organoids as model system in biomedicine, which will be presented by Dr. Isabel Palacios. So before I invite the other speaker, just give me the privilege to welcome our guest of honors who came to join us to learn about this interesting topic. So it is my pleasure to welcome the principal of College of Medicine and Health Science, University of Rwanda, Professor Abraham Mitike. Also, I'd like to welcome Dr. Eugene Mutimura, the Executive Secretary of National Council for Science and Technology. It is our pleasure to be with Professor Muvunyi Claude, the Director of the DG of Rwanda Biomedical Center. Also, we are privileged to have Dr. Sekomo, the Director General of NIRDA. Also, I welcome Professor Emile Bienvenu, the DG of FDA. And lastly but not least, I would like to welcome Professor Mutesa, the Professor of Human Genetic, College of Science and College of Medicine and Health Science, and University of Rwanda. So we welcome all of you to learn together during this interesting seminar. So without delaying, I would like to request Dr. Diane Umuhoza from Inst to give a profile of the invited guest speaker, who is Dr. Isabel Palacios. Who is she? We will be glad to hear her profile so that we can, while knowing who is giving us the information. Thank you.
Thank you.
Is Diane around? Okay. If Diane is not around, then I would like to ask someone else to give this brief introduction of the background of Dr. Isabel. Can I get someone, Mrs. Adedeji?
I think Miss Hassan is there. She's going to read it for us. Hello?
Oh, okay. Yes.
Can you hear me, please?
Yes.
Yes, we
Miss
can.
Hassan Modeno can do that for us.
Okay, that is great. she's welcomed to give us the background and the professional background of.
I can't hear anything. Is anyone talking?
Yes, I would like to confirm if we can maybe hold on for about 5 or 10 minutes to allow more people to join. I can see that we are just about 7 in the meeting room.
Okay.
Maybe we allow for some time for people to join. Thank you. Maybe 5 or 10 minutes for more people to join the room. Thank you.
5 minutes because
Yeah.
I'm traveling to China today. I have quite a busy morning to pack, so I'll do 5 minutes, and then we start, and people can join, even if it's started, I guess.
All right.
Thank you. Hello.
Hello, I'm here.
Maybe while we're still waiting for maybe now we are remaining with 3 minutes. We can start introducing ourselves, some of us, and please put a video on, introduce yourself while we are still waiting, so that like the platform keep having some activity going on.
I think that will take too long, Kushida. So why don't you just introduce me? Even if people are not there, that's fine. They can check online. That's not that important. And we use and then we start straight away after that. I think that's better.
So let me just thank you for your inputs. Due to time, which is not on our side, I can just
because people
the
are
profile.
watching directly from the YouTube. People are
Oh.
already at the YouTube. We are the only one that will be here. Other people are at the YouTube end and they are watching directly from there. Please, we can move ahead.
Okay, so I can just give a profile background of our key speaker.
Yeah.
Then,
Yes.
okay, so thank you very much. Um, actually, Dr. Isabelle is an esteemed researcher and educator specialized in cell and tissue biophysics. She has over 12 years of research experience at the University of Cambridge, and she is currently serving as a lecturer at Queen Mary University, London. Oh, she has made a significant contribution to the science and the understanding fundamental cell biophysical mechanisms which are related to health and diseases. The main research focus of Dr. Isabel is just on, she mainly focuses on eradicating, elucidating cytoskeletal forces and protein function during germline development, employing a multidisciplinary approach integrating cell biology, development, genetics, and biophysics. Dr. Isabel has secured different grants from esteemed institutions such as BBSRC, Wellcome Trust, CRUK, and Alzheimer's Research UK to support those research that she's carrying on. Dr. Isabelle has completed her PhD at the European Molecular Biology Laboratory, Heidelberg. Also, she held various research positions at the University of Cambridge, including Senior Research Fellow and Royal Society University Fellow. She's an experienced lecturer and she's a mentor to undergraduate and graduate students. She has done different outreach services. She's a founder of DrosAfrica, also, DrosAfrica, which is a charity aimed to establish African research community in biomedical science using Drosophila as model system. She had different awards and recognitions, such as the MRC Suffrage Science Award. Also, she's a fellow of Higher Academy, which describes her experience in teaching. Dr. Isabelle has different publications and presentations, and here, like what you are going to share with us based on our experience, we are going to run a lot. Dr. Isabelle, the floor is yours. Thank you.
Thank you very much. Thank you, Kushida. Heck, thank you, everyone else that is joining us today. I wanted to give you an overview of what scientists are doing with Drosophila as a model system, and also what scientists are doing with human cells as a model system. And then I also want to bring this to, or link it to DrosAfrica, which is the charity that I founded with Marta and Lola. I'm not going to go into details of anything. It's more a big overview of a lot of different topics. Some of them you may not have been exposed to very often. And then if you're interested in exploring more any of the topics that I talk about, you can contact me and we can have any discussions. So in general, I have been using Drosophila for many, many years for my research and also for outreach activities, like Kushida said. And now in the last year and a half or so, we have started to work with human organoids. And I will introduce to you what human organoids are, what can they be used for, what people are doing, and also what our lab is trying to do. And the key here is that everything we are trying to do in the human cells are lessons that we learn from Drosophila, and that now we are exploring in human cells. And I'll tell you why the benefits of that combination, what are those benefits, those from fly to humans. And of course, as I said, not just in research, but also in outreach. And just to tell you that some of the work that I present from the lab was done by several of my postdocs and PhD students and also undergrad students. And these are my collaborators and the funding, like Kushida already mentioned. And I want first to thank Inst Africa for this invitation, and also for, congratulate you in getting together a really, really good consortium or collaboration, which is not often seen in Africa, which is across different institutions in the country. So there's a good network and collaboration with common interest as well as individual interest. And I'm very proud of what you guys are doing. So I am following up a talk that Lola, Maria Dolores Martin Bermudo, her name is a bit cut, sorry, gave already on how Drosophila helps the study of human biomedical question and human diseases in general. So I will not talk a lot about that because she gave a great talk on that already. I will touch in a few topics. But again, if you want more discussions on what Lola talked about or what I talk about now, let us know. So as you already heard from Lola, Drosophila melanogaster, which is a very small fruit fly that you may see at your home if you leave some fruits outside, and that looks like that when you look at it a bit closer up, has helped tremendously to understand in general how bodies work, how cells work, how tissues work, how our different systems work, and also as a consequence of that, how we may treat human diseases. So understand how bodies work in a normal situation, and also understand what happens when the body is not working in the normal situation. And you may think, oh, flies, how different are they from us? And of course, there are big differences, but there are also a lot of similarities. These similarities are from body organization, animal development, the development of the different systems, nervous system, digestive system, all the way to the genes that are involved in these systems functioning properly. It's quite impressive how similar we are, in fact. And the thing that Drosophila, I think, is most powerful about regarding research, and that is one of the reasons why I think Africa can benefit from it, is that it's very easy to work with, and it's quite inexpensive compared to mice, compared to tissue culture. Organoids, for example, are expensive, and they are more difficult technically to work with. Flies are easy to learn, and they are very inexpensive to culture and to maintain. And they're also very easy to pack, lots of different mutant stocks you can have in your shelves. And they're also very fast to work with, very, very fast. So you go from adults to a new generation in 10 days. And again, you can do quite a lot and impacting research with not a lot of technology. Of course, you can also develop your lab more if you have the funds and go all the way to, let's say, super resolution microscopy using Drosophila. But you can also do quite or projects that are impacting projects that are not demanding such high technology. And as I mentioned to you, there is a high degree of conservation between human and flies, not just at the level of animal development and at the level of the systems that we have in our bodies, but also the genes, the DNA that is involved in that, and also the genes that are involved in human diseases. Many of them can be found in the fly. And as Lola already introduced a lot of examples, I just want to say that there is a lot of work ongoing in using Drosophila as a model for cancer studies. And that goes from, again, finding new tumor suppressors like the Hippo pathway, which was found in fly and now is being confirmed in mammals and in humans to be linked to cancer, to also understanding the behavior of the cells, the tumor cells within the body. Lots of studies on that. There is also lots of studies on toxicity. It's a whole animal, and it's very efficient and very fast to analyze whether a new drug or a new compound may be toxic at the whole animal level to start with. And of course, you have to move on to other animal systems, but it's used massively for toxicity studies. And it's also being used now for a lot of chemical screening and drug discoveries. Again, you can screen lots of compounds with a lot of animals in a relatively short amount of time and in an inexpensive manner. Then you can move on to the next level of the drug screen, of course. And one thing that has developed nicely the last years, not only with Drosophila, but also with zebrafish, which is another model system, is building avatar. So basically recapitulating in an animal exact genome of a patient. And I invite you to look at the work of Ross Kagan's lab on that. So you can basically phenocopy and genocopy a patient in a fly or in a fish. And then you can use that to look at genetic interaction with other genes or drug screen with the idea of then bringing that drug to the patient closer to a personalized medicine with the specific genetic background of that person. So not just the mutation that is related to the disease, but the whole background of what other mutations that patient may have. And as I said, the systems, many of our body systems are relatively similar. Some of them are also different, of course. One of the systems that the fly is helping lots to study is understanding the nervous system, all the way from development of the nervous system to neurodegeneration diseases. We have conserved several of the systems between flies and human. And what the fly is allowing very nicely is to understand the brain or the whole nervous system, both brain and spine, at the very high resolution level, with not just identifying a group of neurons involved in a specific behavior or in a specific sensory system, but also understanding the connection between all the neurons in the body. And that's, for example, known as connectomics in the Jornelia Research Campus in the States, as well as LATIC labs at Cambridge. And that is, at the moment, this is impossible to do in mice or in higher, even in zebrafish. It's just too many neurons. While in the fly, even though it has a huge representation of the different type of neurons, the number of the neurons is much more reduced. So you can approach these type of projects. And just to tell you that regarding neurodegeneration, Drosophila is being used all over the world to study many of the neurodegenerative diseases in humans. And in addition to all that, one aspect and that we are now very interested in developing further is trying to understand the genotype-phenotype interaction. So basically, if you have a mutation in a population, and as you know, there have been great efforts now in sequencing African populations, and there are around 3 million variants that are maybe linked to diseases. How do you now study that link between the variant, the mutation, and the disease, and the cell biological aspect of that disease, and the molecular aspect of that disease? And flies can help a lot to understand that. Either you study the gene fly itself, or you put the human variant in the fly. It's called a humanized fly. And these are other aspects of biology, as well as agriculture interests that the fly have been helping with. Now, I recently also was reading a paper on how they are using Drosophila to understand adaptation to changes in climate. So a huge variability on what you can do with the flies. What I want to tell you a little bit more today is about what we have been using the flies in the lab to understand biophysics. And I wanted to talk about this because this may be a topic that you may not have been exposed to very often, but it's extremely important for many aspects of biomedicine. So what is biophysics? It's basically a new science, a new field, interdisciplinary between biology and physics and mathematics, and is applying approaches or methods or concepts that have been traditionally used just in physics and in mathematics to now study biological phenomena. And it's been shown that a lot of the biological phenomena are relying on these physical entities, and we need to understand this aspect of biology to fully understand how things work. And it covers all scales, from molecules all the way to populations. So there is biophysics of populations as well. And it is basically applying models and theorems from physics to larger systems, larger what is known as non-equilibrium systems. In our lab, we are looking at cells, tissues, and organs. Many other labs are doing that. But as I said, it can go all the way from molecules to populations. And this is not a unique, this is not a one-way avenue that physics, mathematics, and also chemists, that is a bit more traditional, helping biomedicine, but also the other way around. Biomedicine and the questions in biomedicine are helping development new physics and new mathematics. This is just one example of a case of how a very complex physics concept has been shown to be essential for development of mouse embryos, and is for sure going to be also essential for development of the human embryo. And basically, some biophysicists studied how do you go from a mass of cells in the embryo, the morula, all the way to the blastocyst state. And the main, one of the main features in the blastocyst is the formation of this cavity and the formation of two cell populations, the inner cell mass and the trophoectoderm. And this lab showed in 2019, a science paper, that for this morula to blastocyst chain to take place, you need fluid dynamics. You need fluid flow and contractions and pressure, and a lot of physical concepts need to take place. And it's done by something called fracking. It's an aspect of fluid dynamics known as fracking that is essential for mouse embryo development. It's just one example. And in fact, we have been interested in this fluid dynamics for a long time, more intracellular fluid dynamics. So how do things flow in the cytoplasm of the cells? And fluid, the flow of cytoplasmic components and cellular components, as well as tissue flow, have been studied for several years, but it's really not much understood about it yet. It has several functions, all the way from distribution of nutrients to development of embryos and zygotes. Here you have the zygote of the C. elegans, and fluid dynamics of the cytoplasm has been shown to be essential for these early divisions, as well in mouse zygote, across many different scales of what is needed to. And it's not only in animals, there is also an algae, Cara corallina, that has also been shown that flows is essential for this algae to survive and grow. We use Drosophila as a model system to understand the biophysics of tissue development and organ development. And we use specifically the ovaries of the female flies. So flies have two ovaries, and each ovary is just a machine of producing eggs. This is a younger egg, and this is an older egg. This egg is going to be laid, fertilized, and then a new animal develops. And we are looking at how the dynamics of all the components is essential for the development of that embryo, of the egg that needs to be fertilized and the embryo to be developed. You need precise fluid dynamics as the egg is forming. And we've been studying this for several years, and we have several papers on this with physicists. And what we have found out is that a lot of these forces, a lot of these cytoplasmic flows rely on the function of cytoskeleton. And we've been able to link which cytoskeleton is doing what at what point of the development of the tissue. And again, just to introduce you a little bit, what are cytoskeletons? Maybe you don't get across this very often. They are mainly two major cytoskeletons in cells, the actin and microtubules, but there are also two other intermediate filaments and the spectrin cytoskeleton. Especially spectrin, there is very little known about that. And it's been for a while, or a long time ago, cytoskeletons were known to be important to support the cell and the tissues. But it's clear now that their function is, of course, much bigger than that, going all the way from cell shape, sorry to the typo there, and tissue shape, to, of course, mitosis. And in fact, one of the most efficient drugs in cancer at the moment is a drug that targets microtubules. It's paclitaxel-based drugs, and is targeting microtubules and is affecting many aspects of the microtubules of the tumor cells. And it's the most efficient one at the moment. But function of microtubules is huge. We concentrate on active microtubules and spectrin, and we don't look at intermediate filaments because Drosophila does not have intermediate filaments. That's one of the differences. As I said, there are differences with human. And just to give you a summary of the work of many years, many people, and many papers, we have basically understood quite a lot of the biophysics of cytoskeletons using Drosophila ovaries as a model system, how the forces and the motion are generated, what is responsible for generating what, and what it needs to be moved around, mitochondria, endosomes, and how all this motion and all these forces is actually impacting in the organization, the self-organization of the oocyte, of the egg, which is essential for establishing the anterior, posterior, and dorsal ventral axis of the new animal. So you need asymmetries within the egg that are important for development of the body plan of the animal, and these asymmetries relied on forces and motion generation that rely on cytoskeleton. That's all I wanted to say that is published. What are we now looking at now, and I want to give you a snapshot of what we are doing, is what happens in neurons. Now, if you look at oocytes, and that's also true in mouse, oocytes are huge. There's a big cell. The egg is a big cell. In mammalian, it's a big cell, and in flies, it's a big cell, round, and it needs to mix things a lot because you're going to have to do a lot of divisions. What happens in neurons? Neurons are the other way around. They're very thin and very long, like an axon. We're talking about the axon. But there is a lot of motion required for the function of any neuron. And this is the same in humans. And of course, think of animals like giraffe or elephants. What the length of that axon is one cell, and it has to move things up and down. So we are trying to understand how motion of things, and microtubules specifically in this case, are helping neurons to function. And this is just to tell you how biophysics or physical concepts can help understand motion, not just in oocytes, but also in neurons. This is a pure mathematical paper. I don't need you to understand the details, where they show that also in structures that are cylinders with a radius of around 0.5 micrometer, which is basically what an axon is, you need motion, not just by motor proteins, which are like the cars of a cell, moving on a filamentous, like a microtubule, but also motion by flows. You need many types of motion in cells, also in axons, for those cells and those neurons to function properly. This is just done on paper, mathematically. It hasn't been proven to be the case in neurons, in the real neurons. And that's what we wanted to study. Now, the power of the fly is how much genetics you can do. So we can obtain a fly. In this case, we look at larvae because it's the best system established to look at axonal transport or how things move in axons of neurons. And we can create a larvae that has just a few neurons, and you can actually see the neurons with your own eyes in the microscope because we label them with fluorescent proteins. And we can just mutate a few neurons in the whole animal and then look at the behavior of these mutant neurons in a larvae that is otherwise quite happy moving around. So it's alive. You can look at things happening in vivo, and that's exactly what this movie was. This is transport of material in a live neuron in the larvae that is itself alive. And it allows that to then question a lot of things about axonal transport in the real physiological conditions. And we mutated, and I don't want to go into details of what we did, but we mutated the motor proteins of the microtubule cytoskeleton. These are basically the motors that need to move cargo up and down the microtubule, but also the motors that with this motion are making these flows in the cytoplasm. And what we saw is that when you mutate kinesins, there are two main kinesins in humans and in flies, kinesin 1 and kinesin 3. If you mutate these motor proteins in a group of motor neurons in the Drosophila larvae, these motor neurons die. Compared to control elimination, you get a reduction of number of neurons that are mutant for each of these motors. So basically, it tells you that motor proteins, microtubule motor proteins, are essential for neuronal survival, which is not surprising. Now, what we wanted is then to turn it around. If we need motor neurons for survival of neurons, can we help neurons that are dying by giving them more motor proteins? If we enhance axonal motion, can we help neurodegeneration to slow down? It's, of course, a big question. Can we treat neurodegeneration in patients by making axonal transport healthier, by giving the neurons more motor proteins? So how we did this is to get flies that are expressing a mutant amyloid beta protein, a human amyloid beta protein. So as you know, amyloid beta is linked to Alzheimer's disease and is found in the aggregates that are a hallmark of Alzheimer's disease. It's neuronal aggregates, which are just huge clusters of amyloid beta peptides all together, and they have been linked to the dying neurons and Alzheimer's disease. So we took flies, we expressed in these flies the human amyloid beta mutation that is known, one of the mutations that is known to be linked to these clusters in neurodegeneration. And then you perform a very simple assay, is known as the clambing assay, which is basically measuring the locomotion capacity of these flies. Look how simple we did this. You can do it more sophisticated. There are now ways to do artificial intelligence in looking at how locomotion of flies is happening or is taking place. But you can also just do a very simple assay with empty vials, put a couple of lines. How long does it take for flies to cross one line or the other? Flies like to climb up. If you knock them out, let me show you how we start. You knock them all down, they're going to start climbing up. These are the control flies on the left, and these ones here are the flies expressing the human amyloid beta peptide. And you see how they are unable to climb as well, and to fly also as well. That's because they're having degeneration of neurons, motor neurons, that are essential for locomotion. So what we did then is to ask the question, if I give these motor neurons that are mutant, that are Alzheimer's motor neurons, if I give them more kinesin 1 and more kinesin 3, the microtubule motor proteins, do they perform better? And the answer is yes, they do. So again, here we can see clambing assay quantification of the mutant fly here in red, and the same here. And now if we give to this fly more kinesin 1 or more kinesin 3, again with a genetic trick, we can see here in green there is a rescue of the clambing assay. The flies can do better than the mutant alone. That's true for kinesin 1, and that's true for kinesin 3 too. And they are quite close to control, which is just the normal flies. So you can rescue the locomotion capacity of amyloid beta, human amyloid beta expressing flies, so of neurodegeneration flies, of a model of Alzheimer's flies, but giving them more motor proteins in those motor neurons. We now are collaborating, and this is work with Caroline and Deepthi, which is unpublished. We are now collaborating with the lab of Rick Livesey here in London, and we are trying to ask the question, is this also true for human neurons? And this is how we started getting interested in looking at human neurons. We found things in the fly. We then want to find out whether this is also true in humans. We found them in the fly very easily. Now we want to find out whether it's true in humans. And this is a very complex technique. Again, as I said, much more complex than flies. We use stem cells, induced pluripotent stem cells, to obtain three-dimensional human neuronal networks. And we use stem cells from patients that have amyloid beta mutations. They are either Down syndrome patients, because the Down syndrome have a chromosome 21 in 3 copies, and amyloid beta gene is in that chromosome, so you get more amyloid beta, which results in more amyloid beta peptides, but also familial models of Alzheimer's disease. And Rick Livesey has set up 3-dimensional human networks of stem cells coming from patients. And we are now testing, first, what happened to human neurons if you eliminate kinesin 1 and kinesin 3? This is just to show the expression of the motor. They are there. They are very conserved between flies and humans. What happens to these networks if we reduce the levels of kinesin 1 and kinesin 3? And the answer is they do die, that you have less projection, you have less number of cells. That we've done lots of quantification on that. So as with the fly, kinesin 1 and kinesin 3 are essential for neuronal survival in humans. And now we are trying to understand, is increasing kinesin levels in these patients' human neuronal cultures helping the neuronal survival of those sick neurons? And we don't have the answer yet, but hopefully I can tell you more about that. But just to point out how you can go from a fly question, or from a question that you use fly to respond, all the way to now working with human neurons. Now tell me a little, let me tell you a little bit about DrosAfrica, again, very briefly. DrosAfrica was founded by Lola, which I already introduced you, and Marta and myself. Marta was a postdoc in my lab, and Lola was doing a sabbatical in my lab. We're in Cambridge, and we came up with this idea of helping a little bit research in Africa by bringing Drosophila to the Africa institutions and helping set up Drosophila labs. I don't need to tell you that if you look at a map of population, Africa, of course, is very important, but when you look at the same map with either productions, scientific papers, or enrollment in tertiary education, then of course Africa becomes very small. The same with South Africa, South America and other areas. Compared to Europe, which compared to population, we are very small in population, but we are quite big on university and scientific outcomes. And we thought that we could help a little bit the research in Africa, or this development, by helping build highly skilled community of Drosophilists, starting with, that is also connected. And that's why I'm so happy about the Rwanda Install project, because it's a connected community of scientists. You cannot develop, no one can develop a good impacting project on their own across the world. You need to collaborate, and you need to collaborate, if you're in Africa, with African scientists as much as you can. We are all over Africa, and as you know, you may know, we basically come over for a good 2 weeks of constant interaction, teaching how to run whatever lab of Drosophila you're interested in developing. You may want a cancer lab, or you want a neurodegeneration lab, or you want a behavioral lab, or toxicity, diabetes, and so on. And what I want to say, which is really important, is that the interest for biomedical science and in general for understanding the nature around us and how our bodies work and how they go wrong when they go wrong is a huge bond among participants. And it goes beyond any language, religious, ethnic, or gender barriers. And this is, I think, one of the most powerful things of science. And I hope this stays like that, especially in the current world that we are at with so many conflicts. I just wish science could help more on those, I think, crossing borders. And as I said, we've built a huge amount of collaborators, experts all over the world in whatever you're interested in cancer, in neurodegeneration. And of course, now we are not just, bringing experts from the, Europe or the States. We are really training, helping train African scientists that can then themselves bring those projects to Africa. We've been quite successful collaborating with Amos and the Drosophila Research and Training Center, also with Adedeji in Rwanda now with us, Rashida also in the Inst collaboration. There's many here you can see Adedeji. This was probably our very first workshop ever in Uganda, right, Adedeji? 2013, I may think. Don't know whether he's there, but yes, really long time ago, but really impacting. And you can see we can just use glass bottles to grow. You can grow them just in the open area in your lab. You don't have to have sophisticated incubators, which do help, but they are not necessary. And we've now collaborated with many Africans. And as I was saying, the important thing is to have a connected network. So we don't want just collaboration of Africa scientists with countries outside of Africa, which you can see is growing. We actually want national and African across Africa collaboration, which is very, very flat, and it should be increasing as much as possible. So we have helped create the African Society of Drosophila Research, which we had our first conference just before COVID, and I've heard now that we may be in a good track to organizing the next one, which I'm very happy about. And of course, we couldn't do this with a lot of collaborations all over the world, including Africa, funding from many funding bodies. And join us or help us in any way that you can. Do you have a rich uncle? Tell them about science, tell them about the work of DrosAfrica, and ask them to help us with donations. But especially join us if you're interested. Okay, I'm going to move now. Let me see where we are with time. Okay, I'll do quite fast. Just a look on what is happening in the world of organoids. So up to now, how you could model most of the studies, human studies, was with animals. And to get close to the mammalian study, mouse was, of course, a powerhouse of being used as a model system. But mouse have been shown first to actually be very different from humans, often not bringing much to the table on how understanding humans compared to the studies in other animals. Also, it's very slow, it's very expensive, and it's very difficult to set up an animal house in some of the institutions, for example, in Africa, not just in Africa, even in Spain, but many areas in Africa. So what people have started to explore, and this was a massive development once the field of induced pluripotent stem cells was set up, and I'll tell you a little bit about that, was using cultured organs in the lab that are coming from human stem cells, and these are called organoids. So organoids are basically structures, three-dimensional culture systems that are able to cell organize, that contain many different cell types, and they are functional as close to human organs as possible, but starting from human stem cells to develop those organoids. There is also mice organoids, so you will see work on that, which is also help the fact that you are not using the whole animal, so you reduce animal suffering, which I'm very interested in reducing that. But the most important thing for us here is that you can use human stem cells and develop a lot of organoids that are mimicking very important aspects of the human organ. And even more powerfully, you can obtain the stem cells that you use to create many of the organoids from a patient. And that's very important. So basically, you can get different organoids from pluripotent stem cells. And you can read about that on your own, or get back to me and I tell you where the field of pluripotent stem cell, how it developed. It was a Nobel Prize not that long ago, and by the boss of my husband and a Japanese scientist, John Gordon and Yamanaka. But the power now is that how you get the stem cell, which is by programming an adult like a fibroblast, is actually being obtained from a patient. So you can start with a human that has, let's say, familial Alzheimer's. You obtain a fibroblast from this patient. You reprogram this fibroblast into a stem cell, and then you can now obtain an organoid, brain organoid, if you want that, motor neurons from those stem cells with exactly the genetic background of that patient. And it's been shown that if you start, for example, with an Alzheimer patient, genetically linked Alzheimer's, so familial Alzheimer, go through a stem cell and get motor neurons from this patient, those motor neurons degenerate. They have amyloid beta aggregations, and they have tau tangles in the neurons, so they recapitulate a lot of the diseases of that patient. And then you can do drug screens or many other studies with the neurons obtained from those stem cells that were obtained from that patient. And I want to tell you now that they've been able to basically obtain many different, so the three germ layers, ectoderm, endoderm, and mesoderm from stem cells, which of course allows the development of many systems in your Petri dish in the lab. So they're doing gut, the intestine has been very successful in being recapitulated in the lab. And brain has been very successful, neurons. I'm working with brain organoids in the lab, and I can tell you a little bit more about that. But there are other organoids that people are developing. And let me tell you, just a couple of slides, and you can then go, people can, I'm happy for people to take these slides and share them with the community. So as I said, huge success with intestinal organoids, gut organoids. And in fact, they've been studying now diseases such as cystic fibrosis and looking at differences from the molecules all the way to tissue differences. And they've also been screening drugs for helping with problems in gut and diseased guts. So they are modeling diseases, they are understanding stem cell biology, gut regeneration, cancer in the gut, and so on. They've been very successful with organoids of both endometrial, so female endometrial, and I'm not going to tell you, and placenta, as well as, so endometrial organoids and placenta organoids, which I show here, just by taking some pieces of a placenta. And they have shown now that you can also combine this endometrium with what are known as blastoids, so organoids coming from embryos, and they are trying to recapitulate the implanting of the embryo to your endometrial wall, basically, which is quite powerful. And then they are studying a lot of aspects of endometriosis. They are studying carcinomas. They are understanding just the molecular mechanisms of pregnancy. Just another example. Huge amount of work on that. There's been an effort in developing, as I said, different neuronal tissue-based organoids. It may be cortical, so cortex from your brain cortex. It could be motor neurons, more the spine, the organoids, and so on. Lots of work on that. We are trying to develop brain organoids in the lab with the collaboration with a massive lab here in Cambridge, and it's working quite well. This lab in Cambridge was able to recapitulate microcephaly with these organoids. This is already published. It's the lab of Madeleine Lancaster. She started as a postdoc in Jürgen Knoblich's lab. Now she's here in Cambridge, and she showed that microcephaly in these organoids was related to a very small, for a reduced number of neuronal progeny, so stem cell of neurons, very, very, very early in the development of the brain. So it's a very early defect. As early, let me see if I had the email. I'll show you later. As early as just basically the embryo neuronal tube or neuroepithelia formation, very early stages. Now people are combining type of organoids as well. They are taking, as I told you, maybe an embryo and an endometrium organoid, or they are taking different parts of the brain, or they are taking motor neurons and muscle, and they are trying to get connection between these cell types. So they are generating neuronal networks also, for example, between the gut and the brain. So very impressive field. And this is one example of neurons starting to innervate a skeletal muscle, so muscle. And they have shown that they can induce contraction of the muscle and so on. One weak point in organoids at the moment is the vascularization of those organoids. That's proven difficult. People are trying to get organoids vascularized in different ways, using different type of cell type, but this is still limiting. And of course, that means that the organoids cannot survive for very long. You can carry on giving them nutrients, which you do, and some of the brain organoids survive for months and months. But it would be ideal to have a more in situ vascularization organoid, a blood system that you can then combine with your own organoid. And now what we are looking at the brain cytoskeleton was actually trying to discover or study whether what we found in flies, in this case regarding the spectrum skeleton and the shape of epithelia, if it's true in the human neuroepithelia. I'm going to skip that because it's going through the development of our neuroepithelia, which I don't want to go through. But basically, our neuroepithelia, which is a neural tube that forms all along the animal, and that's going to give rise to everything from the brain to your spinal cord, is basically an epithelia. So the tube, if you do a cross-section of the tube, is an epithelia, which is across just one cell. And in here, you're going to have the neuroblasts, the neuroepithelia stem cells, dividing and giving rise to neurons. And we are studying exactly that. How do you organize these neuroepithelia? How do you organize the cell shape of these neuroepithelia cells? And we found in flies that in epithelia shape in general relies on spectrum skeleton. And now we are trying to understand whether it's true, don't worry about that, whether this is true in our brain organoids, which are shown here. This is stainings that we have in the lab, and we have many of those. We can do 3D organization of these organoids and 3D imaging of these organoids. And we are trying to understand how cell shape in these neuroepithelia, which actually has been also linked to diseases and to the evolution of human brain, how is that regulated by the spectrum skeleton, which we have been shown in flies to affect cell shape of epithelia. Is the spectrum skeleton important for regulating the cell shape of neuroepithelia in human organoids? And the cell shape of neuroepithelia in human organoids, as I told you, is important for evolution of the human brain, is what makes our brain being human, and also for diseases. And we are doing this by CRISPR. CRISPR is another field that we could talk about. We can give a whole talk on that. We are doing CRISPR in interference, which is one type of CRISPR. And CRISPR, I'm not going to go into the details, but it's basically a new way of editing the genome you may have heard about. It was discovered in bacteria. It's an antiviral bacteria kind of immune system. It was discovered in Spain, but then it was translated into human studies and human genome manipulation by Jennifer and Emmanuel that got the Nobel Prize recently for that. And that's all I wanted to say. Thank you very much. I don't want to keep that. And I'm happy to take any questions. Thank you.
Thank you very much, Dr. Isabel, for the great talk. It is indeed a very powerful information to the science and the society. So I'm very sure the audience has a lot of questions.
Yeah.
Ask, and maybe a lot of clarification. So I would like to ask anyone who wants to ask more clarification from Dr. Isabel to raise their hand. Thank you very much. Maybe
Thank you.
Maybe on YouTube, they can just chat on YouTube, and someone will help us to transfer the questions on this platform of Zoom online. Thank you.
Thank you. How do I join the... You mean how do you join DrosAfrica? Yeah, you just write to us, to Lola.
Yeah,
Mark,
someone wrote it.
Yeah, you just write to us and you say that you're interested in joining and helping us, and then you become part of an email group, and we are organizing subgroups, so you can join one of the subgroups. And also, if you have a specific project in mind that you would like to lead within DrosAfrica, then you can also tell us about that. And we just help you develop that project, or you help us develop whatever we are working on. Yeah, it's very simple at this point. We are all scientists that work in DrosAfrica in their spare time, basically, which is very little, as you know. Everyone is very busy. But we are very, how to say, it's very ad hoc. It's a very dynamic situation. Anyone can join, and anyone can bring a project, and anyone can help each other. So we often say, oh, can someone, an email, can someone take over this project? Who would like to join? And so on. So yeah, just write to us. And then you will be part of the website and all that.
Okay. I hope she received the answer. Any more question from the audience?
I didn't see the question. Can someone read the question? Yeah, you can speak. Yeah, talk. I don't know if you
Okay,
Yeah, go ahead.
thank you very much, Prof, for the education and enlightenment. My concern is for you to provide further clarification on organoids and cancer studies,
Yeah.
and as well as the use of Drosophila in rare disease manipulation. Thank you.
Dr. Isabel?
Oh, yeah, I don't know enough about rare disease. It's a good point. I mean, we often, I cannot tell you now with an example of fly with rare disease, but if you do a search, a Google search, and you just use Drosophila as Drosophila and rare disease, then you can first see, is anyone developing a model of fly for a rare disease? Yes or no? But also, if you want to develop a model using the fly for rare disease, you basically have to start, okay, what is the disease? Which system is it influencing? Which gene in humans is linked to that rare disease? Does the fly have that gene? And then you could start by modifying the fly gene and see what happens to kind of recapitulate some aspect of that disease. Or if not, like we did with our Alzheimer's flies, I mean, Alzheimer's in brackets, because the flies, for example, they don't have a cortex, which is where a lot of the neurodegeneration is happening, but it recapitulates a lot of the motor neuron defects. So you can then express the human mutation linked to that rare disease in the fly and characterize the model. Is it recapitulating? Is it a good model? Yes or no? So if you tell me a rare disease and you know what gene it is, we could easily do a search and then find out, is it already being used somewhere? Or can I create it in my lab? So organoids, you also ask about the organoids. Organoids is a bit more of a new system. People are starting to study less rare diseases at this point with organoids. So there is some organoid with mutation of ALS. There are organoids, as I show you, with cystic fibrosis. There are also organoids infected by virus and see what the effects are. For rare diseases, I haven't found yet any work, but it's very new and it's extremely dynamic. It's developing by the day, by the hour. People are developing new ways to grow the stem cells to get different type of that organ recapitulated in the lab. Very dynamic field.
We have some questions in the chat box.
I can see the chat.
Oh, you can't see?
I can.
Oh, okay.
Building a lab for Droso works. How do one start? The first question is, what do you want to study? That's the very first question. Then I would say that we can send you a file. We have a lot of files that we share with our community, with our collaborators, where you have a list of minimum lists that you need. So you need a stereo microscope. You need, if you can get a small incubator, like Marta brought a lot of the flies to Uganda, and they can grow on your bench because they don't need a lot of work. But the problem is that, for example, if you have rats in the university, the rats eat the cotton wool that blocks the vial, and then they eat the fly. The flies escape. They're trying to get to the food. So it's better if it's in an incubator. You know, you don't need a lot of things to start. You need some small microscopes. You need like trays to grow the fly. You need to make the fly food. And then once you know the flies that you want, if they exist in the world, you just write to the scientists, and you could include me or DrosAfrica, Marta, or anyone, even at the DG, someone that is already in the organization. You can include them in the email and say, look, I'm part of this organization. I heard that we could get some flies from you. And the fly community is extremely sharing. If someone has a model system for a type 2 diabetes in the world, they will send it to you if you want to work on that. Yeah. What is the percentage of similarity between Drosophila and human in order to have more reliable results from Drosophila research? So similarity at the genetic level, or what are you wondering? Or similarity at the organization? For example, cytoskeletons are identical. There is no difference between kinesin 1 motor proteins in neurons for humans or motor proteins for in neurons for flies. The difference is, for example, humans have three different kinesin kinesin 1, three different genes, and we had to find out which one is the one expressing motor neurons, so it's more complex. In flies, you often have just one gene for that function. And that, again, makes it easier because you only have to mutate one gene. When you have more than one copy, you have to find out which one is the one expressing those cells. And it's possible that you mutate one of the genes of kinesin 1. They're called KIF5A, KIF5B, and KIF5C. If you mutate KIF5A and you don't see a phenotype, could be because KIF5B is able to take over. You have redundancy in humans or in mammals. So again, the fly is helping understand gene function because you often have only one gene. So how contact DrosAfrica? So basically, I think if you go to DrosAfrica website, let me put it here, or just my email. But if you go to the DrosAfrica website, we should have, we have a contact. Let me put it here in the chat. We have a contact link, but you can also just email any of us. You will have a members list there, and I think you should have the email from everyone. Also from collaborators.
Okay. This is great because many people looks to be interested to join.
Yeah, absolutely.
Not having some ideas of how they can use Drosophiles in
exactly.
biomedicine. Yeah, thank you very much. Any more burning questions? Maybe from YouTube, do you have some questions? Can you look for us?
Yes, those ones are from YouTube. I'm getting them from YouTube.
Ah, wonderful. Good, thank you.
Yeah. So maybe any more questions? We should use our chance to ask now that we have Dr. Isabel with us. Otherwise, yeah, we will maybe use the email. She can share with us the email also in the inbox. Please, doctor, can you give us your email on the chat?
Yeah, but search for anything online because I think everything is available.
Okay. Thank you very much. So,
hello. When you contact us, I just wanted to say, when you contact us with an idea, try to do a bit of homework before. So because it's not that we know everything, we don't know all the model systems or all the model diseases that all the diseases are being modeled in the fly in the world. There's so much with Drosophila. But if you do a bit of research and you find things, you find which lab, we often know them because we meet people in the fly conference and so on, and then we can help you explore. But do a bit of homework before you contact. Just coming and say, oh, what could I do? But no, think about it. What is your interest? What search? Is it already done? Is it easy for me to grow it in the lab and so on, to have the flies? Then you contact us,
Okay, thank you for your input, Sandy. Yeah, it is noted. So if no one has another question?
I think Taufik wanted to say something, no?
Oh, Dr. Tope, please.
Yeah.
Very much. My concern is to confirm procedure for further studies, doctoral programs, postdoc in your lab at the Queen's Mary University in London. How easy is it for us to access the facilities and things?
Yeah, so this is something that every talk that I give, we have this question, not just in my lab, but in general. How can I go abroad? How can I do some studies abroad? And I think maybe we should organize some workshop on this because it's extremely interesting for many scientists, and that happens everywhere. I was in Tunisia not that long ago, and everyone was just wanting to find out because the situation is so bad sometimes. So it's not easy. That's the first thing to say. So, for example, we just put 2 postdoctoral applications, me with another lab, for a postdoc fellowship for 2 Nigerians, and they didn't work out. They just didn't get it. So there are fellowships here and there. There are PhD fellowships. There are more and more, and there are some links. I think at the university level, if you go to the university like Queen Mary, you look for PhD options, and there are PhD now options for minorities from underrepresented groups, and there that you would have links of what may be helping, which calls are existing for PhD students from Africa or postdocs from Africa, but it's not easy. It would be great. I think, yeah, it's true. There should be a website created where all these links are there, the deadlines. You have Commonwealth. It depends on which country you are at. You have some Commonwealth fellowships as well. But it's not easy to navigate the landscape of fellowships and then getting them and so on. But yeah, give it a try. Again, start searching. Maybe we should add in DrosAfrica website a section about this. I tend to share with my connections. You know, Ahmed may have received some from me. When I see a postdoc fellowship that is opening here for Africans or a PhD fellowship, I share with my contacts in Africa and see whether they can then spread the word. I don't know where we could put it so that everyone could see it. Maybe search X, you know, Twitter, the new Twitter, or Facebook if people are posting things there. But yeah, it's difficult. Thank you, Ahmed.
Doctor, please, there is just few more questions. It is like kind of like someone said they normally do toxicology work using rats. Can they grow Drosophila here?
Yeah. So that was one of the examples I put, that there are toxicological studies. And Dr. Adedeji can tell you a lot about that. And Dr. Amos in Nigeria, he's published several toxicological studies using flies. So yeah, it's one of the most developed fields using the fly. There's quite a lot. I don't know, Ahmed, if you want to use anything.
Okay, thank you very much.
So you have a paper on high flux dialyzer. So you need to understand the limitations. So flies don't have blood. So dialyzers for blood, right? Is that what you're thinking about? Or I don't know what high flux dialyzers are. That's for blood dialysis, right? So flies don't have blood per se. They have a circulatory system. It's called the lymph. And they do have a heart that pumps, but they don't have the blood cells. They don't have a blood circulatory system. So blood is difficult.
Mm-hmm.
So you need to know some of the limitations which exist. And again, there are quite a lot of reviews on advantages and disadvantages of using Drosophila for some human studies. What can be recapitulated, what cannot. But sometimes if you are creative, you can develop your own model, even if the gene may not be completely conserved, like we did for expressing human amyloid beta, and then you still get neurodegeneration, even though it's a human peptide that I'm expressing in the fly. I'm not manipulating the fly. So, you know, you may be able to maybe analyze some aspects of heart function, even if they don't have a blood, but they do have a heart. So you can still be creative on what could be done. And again, it's about doing homework, reading what is out there. You could just say, you know, blood studies with Drosophila or blood-like studies.
Okay, thank you very much, Doctor.
Thank you.
This was very wonderful, and
thank you.
We learned a lot.
My pleasure.
So now I think I can invite Benjamin. He's online to give us a vote of thanks. Benjamin, can you hear us?
Yes, I can hear you. Can you hear me?
Thank you, sir.
Yeah.
Okay, thank you.
Thank you. So I'm Benjamin Navana. I work at National Council for Science and Technology as STI Strategic Partnership Specialist. I was given the privilege to give the vote of thanks for the talk today Dr. Isabella has given us. So I would like first of all to thank Dr. Isabella for such an insightful talk that you've given us, learning about how Drosophila can be used to learn human diseases. So thank you so much, Dr. Isabella.
Thank you.
I would like also to thank everyone who participated in today's talk, participants from YouTube and my colleagues here on this line we use. Thank you so much. I would like to thank all the guests we've had, and especially thanking Inst Africa for organizing this. We look forward to more talks like this in the future. Thank you so much.
Thank you very much, Benjamin, for the great talk. Now, as we are close to the end of the session, I would like to ask
Oh, I lost you. I don't know if you lost me, but I have lost you.
Yeah, we've lost the moderator.
Hello.
Hello. I need
Hello?
to go soon. So yeah, tell me about it.
Yes, thank you very much for the beautiful presentation. I think she's back now.
yeah, my internet was unstable. I'm so sorry. It was just time for the closing remarks. I
Yes.
wanted to welcome there's someone to just give us a closing remarks so that we end the sessions. Yes. I was asking Munezero, is Munezero around?
No. I think Prof wants to say something.
Okay.
Hello.
Yeah, I thank you very much. I'm very, very glad to be present here today. I'm very excited. My teacher is Isa. I must make everybody be aware of that, and I'm really proud of her today. The whole of Rwanda seems to be excited with this presentation. It's our first time of coming on here directly on YouTube, and it's live streamed. Very beautiful presentation. We really thank you, Isa. We really thank you so much. I'm excited, and I want to thank everybody who had come online. We'll be having several series coming under Inst Africa, and I wish you a very beautiful afternoon. Thank you very much. Thank you, Isa. We really appreciate you and everybody who has organized.
Thank you very much.
Thank you so much. We really appreciate it. Thank you.
So, as closing, do you have something to tell us at the Deji? Could do that?
No, I'm just corroborating what Prof said to appreciate everyone for attending this program. And we are very grateful to Professor Lola for this inciting talk.
Thank you.
This is fascinating for the Rwandans. I think we are very glad, we are very happy about this. And we hope we move forward with Install Africa. And our next seminar is going to be in June, on the 20th of June. So we hope that we will be more online to witness more of this presentation. Thank you all for your support.
Thank
you, everyone. Take care. I need to go. Thank you very much.
All right. Bye. Thank you. Bye. Thank you. This is a beautiful one. Thank you all for your support. Thank you.